Good afternoon, welcome. I'm David Brainerd from the Department of Psychology, and it's my pleasure to introduce the Pinkel Lecture Series. This is an endowed lecture series with the endowment provided by Sheila Pinkel in honor of her parents, Benjamin and Ann Pinkel. Benjamin Pinkel received his bachelor's degree in electrical engineering right here at Penn in 1930 and had a lifelong interest in the philosophy of the mind, in particular consciousness. And indeed, in 92, he published his own monograph on the topic, Consciousness, Matter, and Energy, and I have a copy here for today's speaker, which you'll receive later. The objective of the book is a re-examination of the mind-body problem in the light of new scientific information, and we use that as sort of the guiding theme for our series. As many of you know, this is our annual event where in cognitive science, interested faculty and students and other colleagues from across the school and the university and the community come, and it's a nice chance for us not only to hear a great lecture, but also to see each other both at the lecture and then talk about it afterwards in the reception, to which you're all invited. That will occur after the talk and the discussion, and will be just up the stairs at a location you came through before the tables were magically installed. So with that background, I'll turn it over to Jay Gottfried, who will introduce today's speaker.
Thank you, David. So I thought I might be able to memorize more or less what I was going to say about Dr. Bargmann this afternoon, but there's just too many accolades that I didn't want to leave out. Before I get into that, I just want to say how excited we all are to have you here today. Dr. Bargmann is a deep thinker and thinks broadly about fundamental questions, not only about worms, but about humans too. She is really a role model, I think, for all of us in terms of trying to understand the mysteries of the brain. So, there you go, your first slide's already up. A few notable facts. Dr. Bargmann received her bachelor's in biochemistry from the University of Georgia. She did her PhD in biology at MIT, followed by a postdoc there with Dr. Robert Horvitz, where I believe you started working with round worms. She took her first faculty position at UCSF. She was there from 1991 to 2004. She moved to Rockefeller in 2004 and is still there as the Torsten N. Wiesel Professor. She was an HHMI investigator between 1995 and 2016. If you've heard about the Brain Initiative, that's basically Dr. Bargmann, who was one of the co-chairs of the advisory committee that brought that all together. She's a fellow of the American Academy of the Arts and Sciences, a member of the National Academy of Sciences. She's collected a few honors and awards over the years. The Lucille P. Markey Award, the Searle Scholar Award, the Takasago Prize for Olfaction Research, the W. Alden Spencer Award for Neuroscience Research, the Charles Judson Herrick Award for Comparative Neurology, the Kavli Prize in Neuroscience in 2012, the Breakthrough Prize in Life Sciences in 2013, and we're only in 2013. Anyhow, this list, I'm sure, strikes you as quite impressive. She has numerous papers in the top-tier journals. I would like to say a few words to our psychology colleagues here who might wonder what we're doing here listening to a talk about worms. I found this quotation, which I think summarizes at least part of Dr. Bargmann's philosophy. She writes, "I study the brain of a worm instead of a human for the same reason that you would want to first study the 1964 Volkswagen Beetle to gain an understanding of how an engine works before you investigate a brand new Boeing 777 aircraft." Recently, Dr. Bargmann took up a post as president of science at the Zuckerberg Initiative, which is a philanthropic effort that began in 2016 to support biomedical research. And one of their key goals for the next who knows how many years is to develop a single cell atlas, a transcriptomic atlas for every cell in the body, not just the brain, but anything down to adipose tissue and maybe even the appendix, for all we know. The vision of this initiative is to solve disease by 2100. Now, except for maybe some of the younger grad students here, we may not be around to see that, but I think Dr. Bargmann aims big, and you'll hear some of that today. So please join me in welcoming Dr. Bargmann.
So I'd like to start by thanking David and Jay very much for the invitation and for the introduction. And I'd like to thank the people I've seen today and will continue to see, and all of you for being part of the conversation. I felt that it was a challenge as a basic geneticist and neuroscientist to talk to a cognitive science and psychology audience. And so I've tried to think about that in today's talk with that in mind. I think I want to start with those framing devices and tell you that one of the things I think that's really interesting about the nervous system is how it's capable of doing so many things on so many different time scales. That some of the activities of molecules might take place in microseconds or milliseconds. That there are things like development and learning that can affect an organism's nervous system for its entire life. But I'm going to focus here on a very interesting zone in the middle, a zone that is of interest to cognitive scientists and psychologists, and that is a zone at which the nervous state generates coherent actions that might be related to one another over the time course of minutes or a few hours, where they can be reliably and flexibly flipped between them into other states, and that give rise to what at a psychological level might be called emotion and motivation, or even at an ethological level behavior, coherent behaviors. So this represents some of the flexibility of the nervous system and its ability to do many different things. And it's this intermediate time scale that I'll focus on today during the talk. And we do have an important insight. Again, you don't want to say that molecules explain the brain or higher order processes, but one of the insights we do have into the roles of molecules as a wedge to drive into these questions of motivation and emotion are the insights that come from human psychology. And those are the fact that drugs of abuse and psychiatric drugs overwhelmingly affect a particular set of molecules within the brain, in fact, that are derived from particular structures within the brain. And these molecules are small molecules. They're not the classical neurotransmitters like glutamate and GABA. They're small molecules like dopamine, serotonin, norepinephrine, and a wide variety of short peptides that fall into conserved families. And these molecules are typically made in structures deep within the brain that project to different areas of the brain. And modulating these molecules gives rise to such robust phenomenology in terms of motivation that it's clear that they do have effects on the way that the brain functions. So they're relevant to sleep, hunger, mood, arousal, social drive, pain, and stress. And understanding what these molecules are doing and what these specialized brain regions are doing seems like it would give rise to thoughts about some of the deeper questions that you all are working on as well. Now, despite knowing about these molecules for many years, despite knowing that things like cocaine or Prozac have robust effects on human behavior, there's a missing element to what we understand about them. So the first thing we know about them is that they tend to couple not to ion channels, but rather to G protein coupled receptors, which means that they act on biochemistry rather than electricity in the nervous system. And biochemistry is something that happens more slowly than an action potential, and it's something that happens on a slightly less defined time scale. So you can't necessarily record a biochemical change with your electrode, and you can't necessarily see the connection between a neuron that releases, say, dopamine and the receptor at the level of an electron micrograph, because you don't have beautiful synapses with pre- and postsynaptic densities associated with them. So there's a level at which we know these things function at a molecular side. There's a level at which we know what these things are doing at a functional side, these deeper functions. But there's a big level in between, getting from point A to point B, that's very difficult to address. So what is the structure of these circuits? What distances do they work over? What timescales? What's their relationship to synaptic circuits? And when I started in this field, people were kind of in one of two areas. Either they were sort of co-transmitter at a synapse, they were kind of mother's little helper, just acting very, very locally, or people thought of them as sort of sprinkler systems was the phrase that was used at the time, that they might be just sort of generally changing the tone of the whole brain, and that was how they affected behavior somehow. And I'd like to try and address some of those questions, recognizing that these are really hard questions to address when you're trying to go from one of the 100 trillion synapses in a human brain to human cognition and behavior. And it's really that motivation. It's being able to work through some of these questions at intermediate times, at intermediate scales, that leads to a simpler organism. And the simpler organism, in my case, is the nematode worm Caenorhabditis elegans. And the thing that makes it really attractive, in addition to its powerful genetics, its tractability, the ease of growing it in the lab, is the fact that its nervous system is completely described. So you have an entire animal with 302 neurons, every one of which is shown on this slide, albeit illegibly. And it's known from the work of John White and his colleagues what the connections are between those neurons. They're the chemical synapses defined at the EM level, the gap junctions defined at the EM level. The overall organization is known with sensory neurons. Here in red, kind of at the top, information comes in, gets processed through a bunch of integrating neurons in blue in the middle, and then flows out through different motor neurons at various locations with a little separate nervous system associated with feeding, like an enteric nervous system off to the side here. So this information is all available, and furthermore, this is sort of a common resource for the whole field. So I know that if I go and I say that I'm going to study the ALM neuron, I know exactly where that neuron is, I know what genes it expresses, I know that if someone else studies that neuron, they're studying exactly the same neuron, and so we can build on each other's work to develop an increasingly sophisticated understanding of what's going on in this nervous system. And from the perspective of the talk I want to give, this nervous system has, in addition to glutamate and GABA and acetylcholine, dopamine, serotonin, molecules related to norepinephrine, and a variety of different neuropeptides. And these molecules and their receptors are quite recognizably similar, homologous to the molecules that might be found in a mammalian nervous system or in a human. So what does it mean to say the molecules are the same and that the nervous systems are the same? Is that anything other than sort of a trivial biochemical relationship, or could there be something deeper about behavior that represents? We think that there could be something somewhat deeper to think about here. So first of all, there is that question of just the biochemistry and how that affects circuit function. But in addition, there's a behavioral relationship that we think is promising to think of as a framework for deeper thought. And to give you an example of that, I'll talk about perhaps the most famous neuropeptide, which is oxytocin, a mammalian neuropeptide first discovered in sheep, where it was discovered that when sheep give birth, oxytocin is flooded into the bloodstream. It causes the uterus to contract, allowing the animals, the pups, to be born. It causes the milk letdown response so that the mother can lactate now and feed her young. And in addition, oxytocin release in the brain of these female animals gives rise to a set of behavioral changes. For example, it's critical in mother-pup bonding. Now, it's not exclusive to lactating or females giving birth. It's present in the brains of all mammals, of all sexes and at all times, albeit at lower levels. And there's also a very closely related peptide called vasopressin that is involved in a set of related behaviors, including social recognition and pair bonding and aggression. So here we have molecules that are related in meaningful ways, both to human medicine. There's probably some women in this room who've had Pitocin to induce labor when they were in pregnancy, but also with a biochemistry and a biology that indicates a very strong relationship to a set of compelling behaviors. Again, mediated through G-protein coupled receptors. Now, this idea is so attractive that it has basically invaded popular culture. So you can get oxytocin candies to give to your sweetheart on Valentine's Day. You can, on the internet, order instant trust oxytocin spray to beam warm thoughts into your partner's brain. You can have clinically proven, doctor-recommended oxytocin factors to improve your relationships. There is an actual book written about this molecule entitled The Moral Molecule, the Source of Love and Prosperity. Can I remind you that this molecule was discovered in a sheep, okay? And in addition, I would like to point out that there is oxytocin, or a molecule extremely closely related to it, in the nervous system of the nematode worm Caenorhabditis elegans, and a lot of organisms with much worse morals than even ungulates. So there's something about this that's obviously ridiculous, and yet there's something a little deeper about this. And the deeper element is the sense that people have that there is something biological or perhaps coherent about some of these motivations that they feel, that they're not purely experience-dependent or learned responses, but that they relate to something that might, in fact, represent something with a biological basis. So what can we say about C. elegans? We can say that by studying this single molecule, which we can eliminate, we can identify where it's found, and it turns out that it's expressed primarily in a single neuron in the tail of a male, and its receptors are expressed in a variety of different neurons in the tail of the male, not in the hermaphrodite, which is basically a female. And these molecules turn out to be important in driving the sequence and timing of male mating behavior. So when Jennifer Garrison was a postdoc in my lab, she studied this peptide and its receptors, their biochemical relationships, and their biological functions. And what she found is that while normal mating behavior involves a set of very well organized steps in a very well directed sequence, animals that lacked oxytocin failed these steps, reversed them, would get stuck and fall off and start over again. All these red arrows indicate the things that went wrong when oxytocin signaling was not present. So there was something about the overall organization of this behavior over longer periods of time, not the individual actions, which actually looked pretty much okay, but rather their sequence and timing that was being affected by the role of this neuromodulator. And so what else can we say? We can say something about the functions at a behavioral level. We can also look at that single neuron in a single animal while mating is going on. This is a C. elegans mating pair. The smaller, thinner one is the male. And there's a little fluorescent structure in the tail of this male right here that I want you to focus on just to sort of orient yourself to where you are in the male as the male starts his mating behaviors. And the male has identified a possible mating partner. He's moving around. He's exploring the surface to try to identify what may be going on. And as he reaches near the vulva, suddenly you start to see a second spot of fluorescence, not the original one, but another one up here. And then as he returns to it and identifies it more firmly, that spot is actually now so bright that it's actually the same brightness as this other thing. This is the oxytocin-producing neuron. This neuron is activated in association with the mating behavior. And you can see that the male is doing a bunch of actions, slipping back and forth, moving around. The neuron stays active throughout the sequence. It's going to do a number of other things. He sort of loses the plot a little bit here. Its fluorescence goes down again. This is a genetically encoded calcium indicator. So we're watching the activity of the neuron in this behavior. We can track movies like this one and ask when it's active and when it isn't. And what we see is that these neurons are, first of all, not only required for mating behavior, but they are active in association with this complex behavior. They're quite dynamic in terms of going up and down. But there are two things that are pretty clear about it. One is that their activity is very low outside of this coherent behavioral state. And that then within this behavioral state where they show this dynamics, they're actually active for a couple of minutes, actually, which is an unusual pattern of activity for a worm neuron and for a mammalian neuron as well. So they're holding activity in a different kind of time scale than some of the other neurons that we might normally be thinking about that might, again, be signaling for milliseconds and then moving on. So that gives us sort of a beginning of an understanding, a deeper understanding of what we can say about this. So first we can say that, you know, this is just a molecule, it could be any molecule, but it looks like there's something that stretches across evolution that keeps oxytocin associated with reproductive behavior in many different animals. And work in other labs has seen similar things. For example, Bill Kristen's lab studying mating in leeches has also found really important roles for oxytocin-related behaviors. And at some point, you start to see them in enough different invertebrates and vertebrates that you start to think that this molecule and this critical biological survival function have gotten linked together sometimes a substantial period ago in evolution and have tended to remain with each other throughout that time. So that's a first conclusion. But then in addition, we have a sense of how it might be doing that give us a little bit more precision in our answer to this general question than just the idea that we go from a molecule to something very complex. Now, obviously, the complexity and the specifics around the behaviors of these different animals are very, very different. But the idea that this is helping to establish that behavioral state may actually be shared across them. So what else can we say about the roles of modulators? I'm going to turn now to another modulator that affects social behavior in C. elegans. Mating is one form of social behavior, but actually most animals have a variety of other social behaviors in association with other members of their species that are not just mating behaviors. So herding behaviors or schooling behaviors, long-term migration behaviors, and so forth involve behaviors that groups of animals carry out together. And C. elegans has an aggregation behavior that also seems to represent some fairly robust response to other individuals that occurs even when they're all of one sex, that doesn't seem to be mating related. And my lab and a number of other labs have been studying this behavior for a long time. And one of the things that we've learned is that these behaviors are potently regulated by a neuropeptide system, including a particular neuropeptide receptor called NPR1. So this is sort of the anti-oxytocin receptor. This is a molecule that when it is active, stimulates solitary behavior, and when it is inactive, releases this form of aggregation or social behavior. And we believe that ordinarily the transitions between activity and inactivity are regulated by the peptides, and in particular by their release by stress. Although the activity of the receptors can also vary in different wild isolates. So for example, this particular British strain that we use a lot in the lab is solitary. Insert Brexit joke here. Whereas the Hawaiian strain shows much more robust aggregation behavior. So again, we have a peptide. And we know some things about this, like we know that this behavior is very strongly elicited by pheromones. So the way that the animals gather together here is by releasing and detecting a set of small molecule pheromones. So we can turn to the wiring diagram, and we can say, hey, the neuron that senses the pheromones, here they are, is called ADL, and that's over here in this map. And then the neuron that expresses the neuropeptide receptor, in which it's required for its action, is down here. It's one of the blue neurons in the middle part of this map. And so how is it that this system is actually affecting these different behaviors? How are you getting either solitary or aggregation behavior out of this? And we can blow up this particular part of the nervous system, and we can study and look at the functions of each step along the path. Are the pheromones present? What do they do to the olfactory pheromone sensing neurons? What happens to the rest of the circuit, and what behavior results? And what we learned from this, and this was the work of Hien Zhang, a graduate student in the lab, is that we have this C. elegans wiring diagram. And within that wiring diagram actually are synapses that connect this olfactory neuron to the two opposite behaviors. They are permanently engaged. They are readable off of the EM structures. There's a gap junction circuit that is a strong driver of aggregation, and that really drives the normal function of these pheromones in a particular set of circumstances. However, when the neuropeptide receptor is active, the gap junction circuit is repressed, and that uncovers a weaker but perfectly functional repulsion circuit that drives the animals apart from one another and gives rise to disaggregation or solitary behavior. So we can say a couple of things about this. So the first is that it gives us a pretty clear hypothesis for what the neuropeptides are doing. And that is the neuropeptides are taking a set of connections that exist in all animals and sculpting them to strengthen some and weaken others, and therefore reroute information flow in a dynamic pattern. So that the difference between this attraction and repulsion represent, again, a rapid and reversible change in the strengths of these different connections that can then allow different behaviors to take place. And then something that's sort of comforting to me from a ego preservation point of view, is that we've had this wiring diagram for over 30 years, and we still can't read it, right? I mean, it's embarrassing. And it's not that complicated. And so now we can kind of look at it and say, well, no wonder we can't read it, because both of these behaviors are encoded in that wiring diagram. You can only know which one is going to be present based on the presence or absence of signaling through this neuropeptide receptor system, which does not show up in the wiring diagram. It's independent of that. And that additional piece of information is there to help you know how the wiring diagram is working at any given time. And these ideas you sort of emerged in the clear view from that point. I'm now going to take you through a couple of other examples of behavior that make us believe that these sorts of behavioral patterns are actually associated with many different kinds of neuropeptides and neuromodulators, and that they act over a variety of different time scales. But again, following the basic principles that I outlined for you there in those social behaviors. I'd like to introduce the next part of my talk talking about the longer-term structure of behaviors and spontaneous behaviors. So I showed you there the behaviors of animals with respect to mating partners, with respect to each other, with respect to pheromones, and so forth. That falls very much in the classical neuroscience formulation of giving a sensory cue and giving rise to a behavior. But if what you really want to study is a motivational or an emotional state, that's an internally generated state. It shouldn't require an external stimulus to drive it. And if that's really what you want to study, it would be better to try to remove the external stimuli and study it directly. And in fact, we've known for a long time that there are internally generated behavioral states that we can monitor in many different animals and in people by just monitoring their behavior. And this first slide here comes from the first paper in behavioral genetics, Konopka and Benzer, 1971, that described the PER locus, the controlled circadian rhythm, studied by Amita Sehgal and others, and just looks at spontaneous activity patterns and shows that even in constant darkness, flies cycle between light and dark phases, but that this requires a specific genetic complement, which is the circadian rhythm pathway, which was recently recognized with the Nobel Prize by among others, Amita's postdoc advisor, Mike Young. So there's circadian rhythms as one form of spontaneous behavior. There's other kinds on shorter time scales as well. So here's a paper showing spontaneous home cage behavior in mice. And there's a clear, with these hatches here representing activity levels. And there's a very clear high activity state in mice. It's in the dark, lower activity during the day. But even within that, the behavior isn't continuously high at night and low in the day. There are bouts of high activity and low activity states in either case. And they're not just sleeping and waking. Some of them are what are called quiet restfulness, different kinds of behavioral states that are associated with different levels of activity. And this same kind of idea of very active behaviors punctuated by more quiet states has been observed very widely in the foraging literature. This here is foraging behavior of gulls, showing that sometimes they'll move over very long distances and other times they'll stay in a relatively small area for quite a long period of time before returning. Once you look for this, you see it everywhere, toddlers on a playground, colleagues in a seminar. So it's clear that there's some kind of a structure here that can be internally generated that isn't obviously related to external cues. And so this is the next thing that we set out to study. And this is a behavioral phenomenon characterized many years ago, initially by Manabe Fujiwara when she was a postdoc. And that is that C. elegans in a completely uniform environment, as far as we can tell, where we give it a uniform bacterial lawn to move on, will show these two rather different locomotion patterns that are called roaming and dwelling. Roaming is a high activity state. The animal moves fast. It changes direction infrequently, and that means it covers a lot of territory in a short time. That's what's in red. And dwelling is a low activity state. It changes direction very frequently, sort of rocking back and forth, so it's awake. Moves very slowly and stays in one area, sometimes for maybe 30 minutes before a roaming bout will make it take off and go somewhere new. So these behaviors have a characteristic time scale and structure. They seem to be truly spontaneous. Similar phenomena, again, have been observed in many different animals. If this were a fly, these might be called roving and sitting, for example, same general structure. And so Steve Flavel, a postdoc in my laboratory, decided to set out to ask, What is it that affects these behavioral states? And specifically what he was interested in was not the individual components like speed or changing direction, but these long durations. What is it that allows these behaviors to have these characteristic behavioral structures? And what he identified after examining a few hundred genes were two different neuromodulators that determine the duration of these two different behavioral states. So one of them is the small molecule serotonin, and that seems to be an important contributor to the dwelling state, the quieter state. And the other one is PDF, pigment dispersing factor. It's actually part of the circadian rhythm system in Drosophila. Its mammalian homologue is called VIP, vasoactive intestinal peptide. Again, very conserved molecules on both sides. And it has the reciprocal effect. It seems to be required to stabilize the ROM states. So if you have an animal that lacks serotonin, dwelling states are shorter, roaming states are much longer. If you have an animal that lacks PDF, dwelling states are longer, roaming states exist, but they're very short. And the substructure of these behaviors is okay. These really seem to be things that stabilize or increase the duration of these behaviors. So what we have here is an idea where we have two antagonistic neuromodulators, and that they're each stabilizing an alternative circuit state. And the animal can flip back and forth between dwelling states and roaming states, in each case using one modulator to extend the one and inhibit the other. So it's sort of a flip-flop of stable behavioral outcomes. Okay, that tells us something about what a motivational state looks like in these animals at a molecular level, perhaps. What does it look like at a circuit level? So from work from many labs, the foraging circuit that gives rise to the roaming and dwelling behaviors, various kinds of forward and backward movements and head oscillations and turns, has been characterized. We can pull out a chunk of that wiring diagram and say, this is the foraging circuit. Now, it's the same circuit in roaming and dwelling. It's just that, for example, the relative balance between forward movement and the speed of forward movement are different in the two states. But it's not like they're different motor neurons involved in those outcomes, for example. And what we can do here is, again, we know exactly which neurons in the worm produce serotonin. Every single one of them has been described. And in fact, 3 of them are directly connected to the neurons in the foraging circuit, here sort of at a high level with the sensory neurons and interneurons. And we don't have to take that for granted. We have, Steve made use of genetic reagents in which he could knock out or reintroduce serotonin in each of these neurons individually, and he could ask, where is serotonin required to give rise to this behavior? Initially focusing on these guys here. And the value of doing this experiment is that these guys here have nothing whatsoever to do with roaming and dwelling behavior. In fact, it's controlled entirely by 2 of the least likely neurons from an anatomical point of view. A neuron in the pharynx within the enteric nervous system that seems to be registering something about food quality makes serotonin. It's not directly connected to a single neuron in the foraging circuit, but it is the most important source of serotonin. And secondarily, a motor neuron, again in the periphery that has only a very indirect connection at a synaptic level, are the key neurons that promote dwelling. So this is a very different logic. It's not sensory top-down, it's somehow inside-out or bottom-up. It doesn't match the wiring diagram. All these connections from the wiring diagram just laugh at the serotonin connections, which are defined by knowing where the receptor is, of course, as well as where the serotonin is, because Steve mapped them both. And he also mapped the sources of PDF, the molecule that supports roaming behavior. Those 2 are sort of far down in the circuit. They are not directly connected by synaptic connections to their targets defined by the expression of the receptors. And what you see when you look at all of this is that this is really a separate logic from the wiring diagram that we see by anatomy. It's a wireless diagram, and it's a distributed diagram with multiple sources of each modulator, multiple targets of each. These cells are not particularly connected to each other. They don't necessarily, except in one case, converge on the same site. And so this logic is really quite different. And we have to think about the modulatory state as being something that is orthogonal to the main wiring diagram. A couple more quick points, just sort of hitting a few things that are actually quite similar to some of the conclusions from oxytocin. First of all, when are these modulatory neurons active? Like the neuron that makes oxytocin, the main source of serotonin becomes active in association with a transition from roaming and dwelling. So at the state transition, this neuron becomes active, consistent with it being required for a long-term dwelling state. And it has a long-term pattern of activity. It's active for about 90 seconds at a time. So again, a long-term behavior is associated with a special class of neuron with a long-term activity pattern. And optogenetics also indicate that this neuron has an instructive role in mediating that transition, because after all, it could have been an effect rather than a cause. But if Steve either imitated serotonin in animals that were dwelling, or antagonized serotonin in animals that were, it's imitated serotonin in animals that were roaming, or antagonized serotonin in animals that were dwelling, he could shift the animals from one behavioral state to another one. So again, in each case, consistent with the idea that serotonin could be instructive in both directions in terms of mediating roam and dwell transitions, depending on whether it was high or low. So after Steve left the lab to start his own lab at MIT, another postdoc joined the lab. This is Shai Stern, and he decided there was no reason to stop at looking at spontaneous behaviors that just happened over an hour or two. And he set up within the laboratory a much more complex tracking system, which is called the black box because it is a black box. It's a very well-controlled environment where temperature, humidity, and illumination are as homogeneous as we can make them. Individual animals, so they don't interact with each other, are placed within individual recording channels and followed from the time that they're eggs to the time that they're adults. And we monitor their spontaneous behavior throughout this entire period. So some people in the lab call this the TARDIS. Other people call it the panopticon. But in any case, what it enables us to do is to track behavior at high resolution. And so what Shai discovered from this is that spontaneous behavior is not just structured into roaming and dwelling over short time periods. It's not just structured into circadian rhythms, which actually worms don't have so much of anyhow. Spontaneous behavior is structured over development. And spontaneous behavior patterns are different in animals of different developmental stages in very characteristic ways. There are 4 larval stages in C. elegans before the adult. This shows the behavior of about 100 different animals. And the heat map is just showing you something about how active they are, how much of the time they're spending roaming. And what you can see is that each larval stage has a very characteristic distribution of roaming and dwelling states. So for example, in the 3rd larval states, the animals are very active at the beginning of the state, lots of roaming, but then they're almost exclusively dwelling at the last half. And these spontaneous activity states are generated over hours. And Shai has examined that in some molecular detail as well. And what he finds is that sort of 2 things. One thing is sort of just a reinforcement of what we already know. So we know, for example, that serotonin is required for dwelling. And sure enough, animals that lack serotonin, showed here, show a lot more of these hot colors than the wild-type animals because it's required for dwelling at every single developmental stage. But you can kind of tell that it still shows some of the normal structure. So for example, it's still trying to roam a lot more in the first half of the 3rd larval stage than in the last half, although it's not regulating it correctly. But Shai identified a couple of other mutants that had also affected roaming and dwelling, but they were specifically required during particular developmental stages in ways that were not characteristic of serotonin. So an example here is that, let's just start with this TGF-beta related molecule. It is pretty much normal in the 2nd larval stage, but at later larval stages, it's quite abnormal in its roaming and dwelling structure. And this neuropeptide receptor here, like the serotonin mutant, is very active at a lot of times, but the temporal structure is different. Most obviously, when wild-type animals hatch, they're very quiet for a few hours, and you see that in the serotonin animals too. Animals that lack this neuropeptide receptor tear out of the eggshell and race around the plate for those first 2 hours. So something about the temporal structure can be regulated independently, but also by modulatory systems. Yeah.
Can you just explain what the x-axis of each one of these plots is and what they each have pockets timeline?
Okay, so every spot on this is a 10-minute interval along the x-axis from the time an egg is laid until the animal is an adult. It's a 55-hour time course. The white lines represent the molting states that separate each stage from the other so that we know with precision that we're separating out developmental stages. And each row represents an individual animal that was tracked throughout that trajectory. And then these blocks here represent different mutant genotypes lacking particular modulatory molecules. So different genetic backgrounds that can give rise to different behaviors. Thanks. So just to conclude, what we think here is that our work is telling us something about the wiring diagram as well as the role of neuromodulators. We think the wiring diagram represents a set of options for behavior, and neuromodulators are helping to select among those options. That the neuromodulatory systems are orthogonal to the fast circuits and can't just be decoded by looking at the wiring diagram. That we see some interesting features, like the source neurons for these modulators have long-lasting activity that may be coupled to the longer activity associated with behavioral states compared to individual actions. They seem to select among the connections, strengthening some and weakening others through mechanisms we still haven't really worked out, but are then rerouting information flow so that, for example, sensory information from pheromones is routed in different directions. They seem to be organizing higher order coordinated circuit states by inducing state transitions and prolonging existing states. And finally, really emphasizing that they operate over time scales that might be minutes for roaming and dwelling, but might be hours for some of these developmental effects. And these ideas are really consonant with work done in a variety of other experimental systems where modulators are also known to be important, where you can sort of extract some of the same features. So the feeding circuits in the lobster studied by Eve Martyr and Brandeis and others, where modulators were important regulators of circuits that rewire their functional activities. The motor circuits of mammals, in particular the dopamine circuits that operate within the striatum and related systems in the basal ganglia, studied by Anne Graybiel, Sten Grillner, and others, also have some of these same ideas about rewiring of activity through dopamine. And even sensory circuits, such as those in the vertebrate retina, where dopamine is used to switch the function of the circuit between dim light circuits that are dominated by rod systems and bright light circuits that are dominated by cone systems. So there's some common ideas across these different states that we can start to integrate with each other and compare. And then just to sort of state pretty clearly what we think the main conclusion of this is, that we think that circuits are overconnected and that we have to understand this yellow modulatory cloud to really see which of those connections are going to be dominating the behavioral output at a given time. And this is part of the C. elegans wiring diagram, but any diagram you ever draw of the vertebrate nervous system, and specifically the human nervous system, will also show you that the brain is overconnected, that brain areas are extensively interconnected with one another, that different regions form, in this case, I think from diffusion tensor imaging, many, many connections, and that we might be able to think of these other systems as ways of strengthening and weakening different subsets of these fast connections to modify behaviors in flexible, reversible, and state-dependent fashion. So at this point, I just want to call out some of the people from my lab at the Rockefeller University who did the work. The work I talked to you about today, a lot of it was done by Steve Flavell, who now has his own lab at MIT in the Brain and Cognitive Science Department. The work at the end about different modulators was done by Shai Stern, who just started his own lab at the Technion in Israel, and his collaborator, Christoph Kirst, who did a lot of quantitative and analytical work in association with this. A variety of different sources have contributed to funding this work. At this point in the talk, these are the people that you acknowledge, and I do want to celebrate their contributions. But I also sometimes feel that I should be acknowledging a set of other people who don't typically appear on these acknowledgement slides. So none of this work would have been possible without the C. elegans wiring diagram, which people just put their nose down and worked on for 10 years to give rise to that amazing EM map. Or without the help of the Genome Project, again, where a group of people worked for about 7 years to really identify all the genes in this organism and their sequences. It was the first animal to have its genome sequenced. And that made possible a variety of studies that just sort of happened in the background of all these things that I told you about. And there are many other tools that come from physics, like lasers, or even from engineering, like microfluidics that came back into biology about 10 years ago, genetically encoded calcium indicators, optogenetics. So much of what we do as scientists is made possible by the tools that people make available to us. And the second thing is that so much of what we do as scientists is made available by the collaborators that we work with. So here I'm calling out Christoph, who did a lot of the analytical work for our long-term behavioral imaging. But I could have, depending on what I've been talking about, have mentioned a long-term collaboration with a chemist, Michael Marletta, or a theoretical neuroscientist, Larry Abbott, where we work together over multiple papers for long periods of time to sort of develop and refine each other's thinking. And this idea that the work that we do as scientists is driven by tools and sort of radiated and illuminated by collaboration was on my mind about 3 years ago when I first met Mark Zuckerberg and Priscilla Chan, shown here with their 2 children. Mark is well known as the founder of Facebook and a software engineer. Priscilla Chan, his wife, is the child of immigrants. She was an elementary school teacher after she finished college, and then she went to medical school. She's a practicing pediatrician. And they decided together on the birth of their first child to give away the majority of their wealth during their lifetime in the service of what they call a future for everyone. Priscilla in particular feels that she's been very, very lucky in her life, and she would like it make it possible for other people to succeed without necessarily having all that luck. Taking luck out of the equation is the way that she puts it, and she thinks that the 3 ways to do that are education, to give every kid a chance, biomedical science, specifically because health is such a great equalizer, and finally, justice and opportunity. So areas like immigration and criminal justice reform are informing. So Priscilla's passions are informing the work in its philanthropy, and Mark's expertise in engineering is informing some of its approaches to how we think there might be fresh approaches to take to some of these problems. So when I met Mark and Priscilla, I asked, they had decided to commit as the first part of their work in biomedical science, $3 billion to science over the next 10 years. And I asked them what it was they were hoping to accomplish through that. And they said, well, we hope that our work will ultimately contribute to supporting basic science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the century. And there's a lot to be said about this phrase here, but I want to say a few things about it to just make it really clear how I unpack this sentence. The first is that if you look back 80 years at where medicine was and you think about what science has accomplished in that time, it is completely astonishing. We are here at Penn, one of the places where gene therapy is being developed, and very specifically one of the places where some of the new advances in cancer therapy have taken place over the past couple of years that are curing diseases that were completely hopeless just a decade ago. But if you reach back 80 years, you're talking about there are no modern antibiotics until about the early 1930s. There is no chemotherapy. We don't know that cigarette smoking causes cancer. We don't know that high blood pressure and high cholesterol give rise to cardiovascular disease. The diseases that we've prevented by changing practice around smoking, by high blood pressure medications, by cholesterol medications, not to mention things like stents and bypass surgery and transplant surgery, have tremendously decreased the death rates from these diseases where even when I was a child, you know, your friend's father would have a heart attack and he would die. That was it. And so I think that saying that science has been able to do a lot for medicine should give us hope that science can continue to do a lot for medicine. And here, this idea that science, basic science, is what will make that possible is also part of the illumination here. Now, still, 80 years seems kind of short, but if you're working with an engineer, maybe it should take 1,000 years. But if you're working with an engineer, he thinks he can make anything go 10 times faster. And so just sort of talking to them, I said, if this is what you really want to do, we have to raise the game for all of science. We have to make all of science better. And I think the way to accelerate biomedical science is to support the development of tools and technologies that will make every scientist a better scientist, and to support open and collaborative models of research that will enable people to work together on problems that are too hard for any one person to solve alone. And I think in neuroscience, we are way ahead of the game in thinking about both of these kinds of problems and these kinds of approaches as key elements to what is going to be necessary for the next generation of progress. So for these first 10 years, we're really focusing on this idea of accelerating science and engaging the scientific community in that acceleration to make things happen. And so we're focusing, first of all, on different ways of supporting collaboration, not just between different kinds of scientists and computational and experimental scientists, but also physicians, patients, engineers, others. Second, developing enabling tools and technologies. And third, we believe pretty strongly that we are a new player in a very healthy, very robust scientific system, and that one of the best things we can do is to help build support for science more broadly, to bring more people into funding science, to help the government, public and private groups, and to sort of engage public support in something that really does have great benefit. So our basic ideas are that we're working through people, technology, collaboration, and open science to advance the field. We have an approach where we work in part as a traditional philanthropy, where we make grants to partners worldwide, typically through open requests for applications that anyone can apply to from our website. But we also have an element of people that come from Silicon Valley through Mark Zuckerberg's contacts, people who come from fields like software engineering and product manager to try to build technology that has advanced more quickly in Silicon Valley in the past decade than it has in nonprofit areas like science and education. So the idea is to bring some of the advances in these fields back into science by bringing some of these expert practitioners in and to collaborate. So the technology group is working in part with our external grants and partners, in part with an internal computational biology group to try to learn on the ground how to best bring these advances and these experimental approaches together to advance the field. We have a fabulous scientific advisory board chaired by Art Levinson, the former CEO of Genentech. It includes people like Harold Varmus, the former director of the NIH, some great people in tech and machine learning, genomics, Bob Tejan, the former head of Howard Hughes. Just to sort of, Huda Zogby, a brilliant physician scientist, just to sort of point out that we know that we're starting something new and that we need to know everything that these people know to help us make good decisions about what to do. And in addition to the sort of senior leadership, we are also viewing our goal as accelerating science, and the only way you're going to find out what's slowing science down is by asking scientists. So identifying what the problems are, what the bottlenecks are, is something that we do by visiting institutions, visiting other philanthropies, the NIH, and holding workshops and hackathons and all kinds of projects where we try to find out from people what we can do that could have a systemic effect on a field and could make a differentiated impact in accelerating science. And so I'll give you a couple of examples. We're just a little over 2 years into our programs, but we're sort of trying to develop these ideas, and I think we're at this point sort of coming up with a point of view about what it is that we can contribute. So for example, one of the ideas we want to do is to accelerate the ways that people can share and discover information from the rest of the scientific community. And we are doing that, for example, by funding the preprint server bioRxiv, which enables people to share their manuscripts with each other prior to publication in a way that at this point all of the major journals in basic science accept as a reasonable thing to do. And why shouldn't they? The physicists have been doing this on arXiv for 30 years. At the same time, we're also building a machine learning tool called Meta that uses artificial intelligence to help researchers discover papers and preprints that are relevant to their own specific interests. So this is personalized and predictive ways of looking into the literature and pointing out to you papers that you might have missed otherwise that could be directly relevant to the experiments that you're doing. So these things are really, these are dry work things. Here we're working with an external group at Cold Spring Harbor. Here it's our own software engineering team that's working on this, but they're really toward common goals. There are other kinds of tools and other kinds of collaboration that we're carrying out. Jay referred briefly to the Human Cell Atlas project. Now, this is really, it's not run by us. It's a grassroots project run by Aviv Regeb at the Broad and Sarah Teichmann at the Wellcome Sanger in the UK. There are several hundred scientific groups around the world who've gotten really fired up about the idea of really building a reference map of the cells in the human body. And this is being powered by advances in single-cell genomics that make it possible to now look at the individual patterns of gene expression in a cell, by sophisticated new imaging techniques that enable you to even look at many hundreds of genes in cells in a tissue and their relationship to each other. And we are here supporting the research groups that are working on developing these technologies and improving them, supporting the research groups now that are starting to assemble the data for a first draft. And in addition, we're helping to build the data platforms in collaboration with the Broad, EBI, and Santa Cruz, the great genomics groups, to bring some of the Silicon Valley practices for working with very large data sets into the biomedical literature that fields that have not been able to have access to them until now. So that's a collaborative project. It's us and other funders. It's us working directly with other software engineers and computational scientists, and it's us funding a variety of scientists. And the scientists are remaining in charge of the intellectual direction of the project. We are really guided by them in this endeavor. Another collaboration so this is we like this because we feel like it's a perfect example of explaining what does it mean to support science that could help you solve all diseases. Every disease has a cellular basis. If you know what all the cells are and what genes they express, you have a basis for understanding every disease more deeply than you could have. It's not dedicated to one project or one lab or one institution. You can study these things very broadly and use this as a foundational resource. It's like the Genome Project, but for cells. And doing this kind of stuff is it's not glamorous. It's pretty fundamental. It's infrastructure. It's a platform. But it enables other people to do their best work in the same way that having a reference map of human genes enables other people to do their best work. We're not afraid of addressing diseases directly. We're just starting our first disease-related project, what we call the Neurodegeneration Challenge Network. Again, where we're trying hard to do things like make it possible for people who've never worked on neurodegeneration to enter this field. By allowing them to do so in a collaborative environment where there are other scientists and physicians and computational scientists who can help them build an interdisciplinary approach. We have an emphasis here on the next generation of scientists. We're particularly interested in supporting people at the assistant professor level in this project, but it's not exclusive, it's just an emphasis. And at this point, between these two projects, we're supporting over 200 groups in 15 different countries. And here's some other things that we're just rolling out this month. We're starting a project where we're supporting imaging and microscopy, both by supporting staff scientists at core facilities, really important people for technology, supporting research, education, dissemination, innovation, kind of underappreciated and underrecognized in our existing academic systems and by our existing grant systems. And we're also now gearing up to start substantially supporting open source software for microscopy, because microscopy is a big chunk of a lot of modern biology. And finally, we just want to think hard about what kinds of people we want in the field. Here we want people in a particular setting, but we would like to see more people in science in general. In particular, we feel that the representation of underrepresented minorities is still not what it should be in science, and so we are helping the University of California schools to clone a very successful model developed at the University of Maryland, Baltimore County, that's done an amazing job of bringing underrepresented groups into science and ultimately into PhDs and faculty positions. So we're trying to think about things a little differently. I appreciate being granted these 10 minutes to tell you what we're doing because we are very open to feedback still. We're still developing our point of view in our programs. We want to hear from you what will make a difference. I do want to say that if we're going to think about science a little differently, as we also have to evaluate it a little differently. So we can't just be saying, the paper you publish in a fancy journal is what determines whether we fund you. We need to reward people for other kinds of productivity, for writing great software, for developing valuable data sets that get reused, for protocols that enable the whole field to move forward. We should be open to funding someone who never publishes a paper because they've generated something else for the scientific community that's valuable. And similarly, we really care about things like, are these things available? You should get as much credit when somebody else publishes your mouse as when you publish your mouse. Are they requested and reused? And finally, if we value collaboration, we should explicitly reward collaboration. We shouldn't say that's sort of a side point, but rather that some of our criteria should include things like leadership, willingness to be a co-author instead of always the senior author, the success of the people you work with, and so forth. So we're really looking for people who share our goals and values. We have positions. The science group at CZI is about 90 people. Of those, 70 are software engineers and in related technical areas, but we also have program managers, computational biologists, people who are focusing on the meetings and convenings and the ways that we get people together to collaborate more and so forth. the tools, the projects, the RFAs are posted on our website and are for the most part completely free and open for applications. And you can also write us directly with your ideas, your thoughts, and your criticisms. Thank you for this time. Thank you for spending this hour with me. And,
we have time for questions. Is this going to be wandering the crowds?
So beautiful. I've been thinking about going back to the part of your talk where you talked about these two different strains of C. elegans. I think they were called British and Hawaiian. And I gathered that they both had the same circuit, and what differentiated them was a peptide and how it triggered. But in terms of the strains themselves, is it the amount of expression of that peptide that differentiates whether they're going to be British or Hawaiian, or is it something else?
It's actually a point mutation in the peptide receptor that changes its basal level of activity so that it is more or less sensitive to the input. So what seems to be the endogenous regulator of this behavior is a stress signal. When animals are at low stress, actually they're all solitary. When stress levels, particularly oxidative stress, are very high, they all aggregate. And the transition point at which that occurs is different between various wild strains because of this. So it's sort of setting the point at which that behavior flips over. So both behaviors are accessible to both strains. It's just the transition that shows a difference.
Cori here.
Hey,
so
Anita.
that was great. So a question I had was, to what extent are the mechanisms shared between the short-term behaviors, the roaming and dwelling, and then the ones that you're looking at through the larval stages? So for instance, does PDF affect activity in the larval stages, and does TGF-beta affect roaming and dwelling?
Yeah, so that's an excellent question, and it is going to be addressed by Shai Stern in his own laboratory. He actually started from a Drosophila developmental background, and so he's very interested in exactly these kinds of questions about different timescales. I mean, my money is on transcriptional mechanisms for these kinds of long-term effects. There's nothing sacred about a neuropeptide or a neuromodulator that says that you can't turn it up or turn it down over particular developmental times. And then at the same time, regulate it more dynamically over short time periods, such as its release. So. And I think, there's a TGF-beta pathway that is, in most circumstances, a transcriptional regulatory pathway, although there are a few examples at synapses where it can operate more acutely.
So maybe like learning and memory, where the transcription is what?
Yeah, that's a great analogy. So you have learning and memory where you have short-term changes that happen at the level of synaptic plasticity. In the classical Eric Kandel case of Priscilla-Gill withdrawal reflex, actually through the action of serotonin that affects presynaptic release, so basically short-term rewiring of the circuit. And then increasingly longer and longer-term activities involve translational regulation, changes in gene expression, structural plasticity, and so forth. Yeah. So biology does not stick to principle. It's opportunistic. Yeah.
So one thing I understand from your talk is that behavior in C. elegans seems to have a fundamentally discrete nature. And this is not the first time I see signs of discrete aspects in behavior. For example, insects also seem to switch between discrete states. Another example that is interesting to me is the cuttlefish, which has a finite discrete number of possible skin patterns. So my question is, do you think that behavior has a discrete nature, and to what extent these extend to animals with more complex brain and less stereotypical behaviors?
This is a great question. I particularly enjoyed the cuttlefish part of that question, where the end organs are actually the color, the chromatophores, so the organisms and not the muscles. I am 100% positive that there is a greater range of behaviors and a greater range of flexibility and manifestation of behaviors in, frankly, any animal with legs compared to a worm. let alone mammals and more complex animals like that. So we are working in a simple animal. It's successful. It's had the same number of years of evolution to be good at being a worm as we have had at being good at being people. So it's a good nervous system, but it does not have the same kinds of diversity of outcomes that ours can have. At the same time, I'm not sure that the emphasis on stereotypy is a true feature of the animal as opposed to something that we impose upon it. So when you are an experimental biologist, what you do is spend a huge amount of your time trying to make things as reproducible as possible. And that's one of the reasons that people love to use sensory stimuli to elicit behavior. And then they want the sensory stimuli to elicit the behavior every single time so that they have something reproducible. And then they say, aha, this behavior is stereotyped. And increasingly, we look at a lot of olfactory behaviors. And a lot of the olfactory behaviors are intrinsically variable and probabilistic. And you can make certain ones of them extremely deterministic by working very hard to control the circumstances. But I think their real nature is in this more probabilistic space. And we can talk for a while about why in foraging strategies that is actually the better strategy. And many of the foraging behaviors we look at are probabilistic. So how certainly the temporal patterns have quite a wide range among them. Again, some of the categorizations in roaming and dwelling encompass a range of subsets within that. There are people in the lab trying to kind of pull those apart at the moment. So I think we shouldn't be too quick to forget how we got to the path of where we are in saying that these behaviors are stereotyped.
But you showed that it's organized into few discrete states.
Yeah, but I defined what those states were. And I didn't show you what went into that. I mean, certainly forward and reverse are different states, but roaming and dwelling, for example, there's a range of speeds that could be roaming. There's a range of reversal rates that could be roaming. They actually differ slightly depending on how much food is around. You can always draw kind of a line between two states, but the shape of those states is actually different. So
It's not discrete.
it is, you can set up conditions in which it is absolutely discrete, but that doesn't mean that it looks that way in the full range of normal conditions. Yeah. Again, it's sort of the experimentalist desire to have it look that way, because that's what we're trained to do.
Thanks for your fantastic talk. I both appreciated the research side of it and the more outward-looking structural side of it. I think it's really important that researchers are engaged in both of these things. And my question is actually related to both pieces. So one of the beautiful things that the research part of your talk showed was that a kind of traditional, discrete, computational approach to C. elegans is not going to get the whole picture because we need to add this chemical dynamical component. And a question, concern, excitement for the future is that we need theory. We don't just need tools. We have no idea how to combine those two perspectives. And so thinking both about the research side of your talk and about the sort of more outward-looking side of your talk, I think it's really important to include a kind of not just tools, which is, I agree, super important, but adding extra support for theory. So I wanted to both invite you to comment on how you think that might go in the specific case that you discussed, and also more generally about including room for more theoretical investigation in the Chan-Zuckerberg initiative.
Yeah.
Thank you.
Yeah, so that's a great question. I'm happy to say that in the BRAIN initiative, largely, Terry Sinofsky was one of the members of the committee that set that up, and we actually sort of highlighted theory as one of the really important aspects of what was going to be necessary for neuroscience. And I think at the time that Terry said, look, we really have to put that in there, that was still a little bit of a surprising thing for people to say 6 years ago. And so, and I think now it's really accepted in the neuroscience community that that's essential. So I think that, so I'm with you. I endorse that statement. I would say that so far at CZI, we do feel that we will probably end up with a fairly strong emphasis on things like computation and analysis and more quantitative approaches as something that the next step of a lot of biomedical science is going to do. We have not been thinking so much about theory qua theory as we have been thinking about large data sets, time series, multimodal integration, how questions like that. But it's sort of a good thing to step back and consider whether that should be something to do more explicitly.
Yeah, so just for
Yeah.
example related to the
Yeah.
last question, making sure that we're choosing our variables appropriately, having a conception of what motivation and behavior is such that we can use these tools in the most effective way possible. That seems to me to be a really important side of the project.
Yeah, and even, even the kind of unglamorous stuff that we think about, it's like, how do you think about things like normalization and harmonization and things like that? Those are, again, they're partly empirical to deal with the data you have in front of you, and partly kind of guided by by an understanding of the underlying quantitative structure of the data and what assumptions you should and should not be making. So they have a little bit of that, but I think you're asking something deeper that we haven't thought about so much. Yeah.
Next question's here. So you showed that that roaming and dwelling was up and down regulated by, if I'm remembering right, the levels of serotonin and PDF. Is that right? So I was just wondering if you push that back a little further and have put any effort into understanding what's up and down regulating the levels of serotonin and PDF.
Yeah, there's something really interesting about serotonin regulation. Again, I get to smile and tell you that Shai is going to work on that in his lab, which is actually the truth, which is, but the serotonin, in work that I didn't tell you about, one of the things that he found is that that individuals vary from one another in a constant environment with a constant genotype, so that some of them are very active and some of them are very inactive in ways that you can't understand. He calls that individuality. And similar things have been seen in flies, actually, where it's called personality, but we don't think worms are persons. And that is dependent on serotonin. If you take away serotonin, or one serotonin receptor, which actually doesn't affect roaming and dwelling as a whole, all of the animals collapse down to acting like the average. So so it says that there's some active process generating variability between individuals. And some hints suggest that that's something about the regulation of the serotonin system, possibly at a transcriptional level that gets kind of locked in at some point and then sustained through development. They're not super strong hints, but there's a lot of, but this is an example of something where, you start with a modulator and then you can have transcriptional regulation of the modulator, you can have potentially environmental regulation, you can have, we think, stochastic regulation that then nonetheless leads to a stable state. So I think that this may be a much more rich area than we're giving it credit for. And I mentioned Eve Marder very briefly. I mean, Eve Marder has shown a really profound result in neuroscience, which is that you can get the same properties of a circuit from different molecular contributions and different weights of synapses and things like that. So the circuit has kind of a destination that it maintains. But one of the important things about that seems to be that you go, if you go through a particular set of states, you can recover from them, but you don't recover by going back to the ground state, but by going through a different state and yet another state. So the regulation of the modulators may actually be itself changing and thereby leading to changes in outcome. And I think that could actually have some interesting implications for some of the things that people have been talking about recently, for example, about long-term use of antidepressants and things like that. Yeah.
I was just wondering what function this was serving for the organism or for the population of organisms.
Yeah, we have sometimes played with setting up competitions, sort of toy evolution experiments, and so we can actually see effects of some of these things that depend, for example, on the animal density and the food distribution and so forth. So the idea that there are multiple behaviors for exploitation of different kinds of environments cannot be excluded, because you can make environments that fairly readily uncover that. Yeah.
Hi. Very cool talk. In your talk, you seem to suggest that the traditional wiring, which is like a spatial connection map, is not enough. You need more like a receptor-ligand interaction, like paracrine, including them to be like in a spatial temporal way to be a more complete map. And my other question is that, do you think like in terms of like the AI research, like people do a lot of neural network and they create certain behaviors, but they can't explain each node in the deep network what they are doing. Is that the case for like researching like the neural network in the C. elegans? Like, do you think we are looking at the right thing, each like signaling molecule, or each cell is the right module we are looking at? Can you comment on that? Thank you.
So the first, I felt like the first thing was not a question, but like, yes, exactly what you said. Wiring diagram is not sufficient. The second question is about artificial intelligence and whether essentially there will come a point at which the sort of features of a neural network and the features of the artificial intelligence network will be informing each other. And Nicole and I were just talking before the seminar about the fact that in mammalian visual systems now, you know, the kinds of models that people have developed for deep learning in object classification, visual recognition, are sort of shedding light on features of the visual processing areas in the mammals, which is not really a huge surprise because those models were derived by Jeff Hinton, who was trying to develop the theory for understanding visual processing in areas like V4. You know, so that does seem to be an area where there is already a dialogue that's helping people understand how those things work. And I think especially in the past couple of years, when people have been making more of an effort to sort of crack open their machine learning networks and really understand how they're working, I think that's required to make that next step. If you just sort of, you know, throw something into it and get something out, then it'll be hard to reach scientific insight. But with that, I think it's possible. Yeah.
The question is related to the prior question. So with the roaming and dwelling spontaneous bouts, they appeared to have like there were distinctions across larval stages in the lengths of time. So my question is, why is that the case? I think L3 had like a longer, forget which one it was. Does that correlate with like a known biological feature in each like developmental stage, or? Yeah,
no idea why L3 has this pattern. So presumably, yeah. I don't know. Correct answer, yes.
I think that's a good place to stop. Thank you very much, Dr. Bargmann.